All right, everyone, welcome. Grab your notes and get settled because today we are diving into a comprehensive masterclass explainer on Chapter 11, Holozoic Nutrition. I am absolutely thrilled to guide you through this one. We're going to be looking closely at exactly how organisms, from microscopic single cells all the way up to complex human beings, take in and process the fuel they need to survive. It's a fascinating biological journey, so let's unlock the secrets of the digestive system together. Okay, let's dive into this. Here is our roadmap for today's lesson. We'll start with the basics of holozoic nutrition, followed by a look at the amoeba and planaria. Then we transition to human digestion, covering both the upper and lower tracts. Finally, we'll wrap up by exploring the vital role of the accessory glands and looking at some common digestive disorders. Starting right off with Section 1, the basics of holozoic nutrition. Think of a living cell kind of like a car. Just as a vehicle needs fuel to move, a cell needs a continuous, steady supply of food to function, grow, and repair itself. Now, holozoic nutrition involves taking in ready-made food and processing it in 5 very sequential, highly organized stages. First up is ingestion, literally just the physical act of taking the food in. Next is digestion, where complex, non-diffusible chunks of food are broken down into simple, soluble molecules. Then comes absorption, where these molecules cross the digestive membranes and enter the body. Now, class, please pay close attention to step 4, assimilation. This is incredibly crucial. It isn't just absorbing nutrients, it is the actual utilization of those absorbed molecules during the cell's internal metabolic processes. It's the putting the food to work phase. And finally, egestion, which is simply the removal of any leftover undigested waste. So how does all this breakdown actually happen? Well, digestion can be intracellular, meaning it happens entirely inside the cell. You typically see this in single-celled organisms that engulf their food into a tiny vacuole, or it can be extracellular, meaning it occurs outside the cells within a gut or cavity, where digestive enzymes are released into the space. This is what we see in most multicellular animals. Furthermore, the breakdown process itself is a 2-part team effort. It's mechanical, like physical chewing or muscular churning, which drastically increases the surface area of the food. And it's chemical, involving specific enzymes that facilitate the hydrolytic breakdown of those food molecules. Basically, mechanical mastication brilliantly sets the stage for chemical digestion. Class, I want you to really know the difference between a sac-like and a tube-like digestive system here. A sac-like system, which we find in lower, simpler organisms, has only a single opening. That one opening serves as both the mouth and the anus, and digestion there is often much more chemical than mechanical. But a tube-like system, like the one we have, that is a massive biological upgrade. It features 2 separate openings, a mouth for entry and an anus for exit. And this setup is a game changer because it supports highly specialized regions with variable pH levels, enormous enzyme diversity, and really strong muscular support for vigorous mechanical digestion. Moving on to Section 2, let's look at some specific examples with the amoeba and the planaria. Let's start small with the microscopic amoeba. Since it's literally a single cell, it uses these temporary cytoplasmic extensions called pseudopodia to trap its microscopic food. It forms a little food cup that pinches off into a food vacuole inside the cell. Then enzymes are pumped straight into this vacuole for intracellular digestion as it circulates through the cytoplasm, a neat process called cyclosis. Once it's done, the waste is just pushed back out via exocytosis. Now contrast that with the planaria, which is a flatworm. It captures prey using a mucus-secreting muscular pharynx and basically sucks it into a highly branched, sac-like intestine. What's cool is that planaria actually starts with extracellular digestion in the intestine cavity, and then the cells lining that intestine engulf the smaller particles to finish the job intracellularly. It's a fascinating hybrid system. All right, Section 3. This is where things get really relatable. Human digestion, specifically starting with the upper tract. I want you to shift your focus inward for a second and imagine the sheer structural complexity of your own gastrointestinal tract. Let's start right at the lips. Inside your mouth, humans typically have 32 adult teeth, heavily specialized for mechanical digestion. You've got 8 incisors at the front, flanked by 4 sharp canines. These are your cutting and biting tools, perfect for piercing and tearing into food. Moving back, you have 8 premolars and 12 mighty molars. These are your heavy-duty grinders and crushers. Working together, they mechanically pulverize your meal, significantly increasing the surface area for enzymes to act upon later. Inside the oral cavity, once you've chewed your food and mixed it thoroughly with saliva, your tongue steps in. It's covered in sensory papillae, and it helps form the chewed food into a soft, manageable ball called a bolus. Next comes deglutition, which is the scientific term for swallowing. This is an incredibly highly coordinated, voluntary process where your tongue pushes the bolus toward the back of the throat. As this happens, a crucial little flap called the epiglottis perfectly drops down to block your larynx. This ensures the food goes down the esophagus and not into your airway. Literally a lifesaver every time you eat. Now, how does that food actually travel down the tube? It's definitely not just gravity doing the work. It's a process called peristalsis. This brilliantly illustrates how involuntary circular and longitudinal muscles coordinate. Circular muscles right behind the bolus contract, while longitudinal muscles ahead of it relax, creating a rhythmic wave that actively pushes the food down your esophagus and through your entire gut. On the flip side, we have anti-peristalsis. This is the reverse squeeze. It's a defense mechanism triggered when the gut is irritated or overdistended, forcefully pushing contents back up. And yes, that is exactly what we experience as nausea and vomiting. So the esophagus delivers that bolus straight into the stomach, which is a J-shaped muscular reservoir situated in the upper left of your abdomen. It is closely guarded by two valves, the cardiac sphincter at the entrance and the pyloric sphincter at the exit. Structurally, the stomach is divided into four main regions: the cardia, the fundus, the main corpus or body, and the pylorus. But what makes the stomach mechanically incredible is its musculature. Most of your digestive gut has circular and longitudinal muscle layers, but the stomach adds a unique inner oblique layer. This third layer allows the stomach to vigorously churn, twist, and mash the bolus, mixing it with gastric juices until it becomes a thick acidic paste known as chyme. Let's zoom into the dense cellular biology lining that stomach, known as the gastric glands. In the fundic region, we have parietal cells. These guys are working overtime, pumping out hydrochloric acid, which drops the stomach's pH to a highly acidic 1.5 to 2.5. They also secrete something called intrinsic factor. Right alongside them are chief cells, which release pepsinogen. Now, pepsinogen is an inactive enzyme, but when it hits that highly acidic HCl, it converts into pepsin, which is the active protein-digesting enzyme. Finally, down in the pyloric region, glands are busy secreting a protective mucus layer, and crucially, the hormone gastrin, which actually acts as the signal to regulate all these highly acidic secretions. Okay, moving right along to section 4, the human lower tract and intestines. We now follow that acidic chyme as it slips through the pyloric sphincter and enters the massive lower tract. The small intestine is a roughly 6-meter-long marvel, and it's divided into 3 distinct parts. First up is the short 30-centimeter duodenum, where that highly acidic chyme is immediately neutralized by bicarbonate from Brenner's glands and mixed with bile and pancreatic juice. Next, we hit the 2.5-meter jejunum. This acts as a chemical powerhouse, secreting specific enzymes like maltase, sucrase, and lactase to break down different disaccharides, along with peptidases for proteins. Finally, the chyme enters the 3.6-meter ileum, which is specially adapted to absorb vitamin B12, bile salts, and any remaining nutrients. Now, what's really interesting about this is how the body maximizes all of that absorption. The small intestine's inner wall isn't just a smooth pipe. It is heavily folded and densely covered in microscopic, finger-like projections called villi. And it doesn't stop there. The cells on the villi have even tinier hair-like structures called microvilli. This drastically increases the surface area. Inside each individual villus is a network of blood capillaries ready to absorb sugars and amino acids directly into the bloodstream. There is also a specialized blind lymph vessel called a lacteal in there, which is specifically designed to absorb and transport fatty acids away to the thoracic duct. So what happens to the leftovers? The undigested matter and water finally pass through the ileocecal valve and into the large intestine. It starts in the cecum, which is where you'll find that vestigial vermiform appendix we've all heard of. From there, the matter travels up, across, and down through the ascending, transverse, descending, and sigmoid colon. This is where water and salts are heavily absorbed back into the body. Crucially, your colon is home to billions of symbiotic bacteria, like E. coli. They thrive there, and in return, they actually synthesize vital vitamins like K and B for us. Finally, the resulting dehydrated feces are stored in the rectum until voluntary and involuntary sphincters coordinate to allow for egestion, or defecation. Which brings us to section 5. To really understand the digestive tube, we have to look at the vital external accessory glands supporting it. Think about your mouth watering when you smell a really spicy or savory food. That reaction is your salivary glands instantly kicking into gear. Humans have three major pairs, the parotid, sublingual, and submandibular glands. And get this, what is truly astonishing is that a normal, healthy person produces 1 to 1.5 liters of saliva every single day. That is enough to fill a large water bottle. This constant fluid lubricates our food with mucin, it jumpstarts starch digestion with the enzyme amylase, and it even acts as a mild antibacterial agent using lysozyme. Next is the liver, which is the largest gland in your entire body. The liver's cells, called hepatocytes, synthesize a greenish fluid called bile. This bile is stored in the gallbladder and is essential for emulsifying fats in the duodenum, basically breaking large fat globs into smaller, manageable droplets. But beyond just digestion, the liver is a massive metabolic factory. It's a major homeostatic regulator. It manages your blood sugar by converting glucose to glycogen, metabolizes fats, and performs deamination. Deamination is where it strips nitrogen from excess amino acids to safely form urea and uric acid. It even synthesizes brand new non-essential amino acids for the body to use. And then we have the pancreas, which is an absolute multitasker. It pulls double duty as both an exocrine and an endocrine gland. As an exocrine gland, it pumps highly potent pancreatic juice into the duodenum. This juice is absolutely packed with digestive enzymes like trypsinogen, chymotrypsin, amylase, and lipase, which together break down proteins, carbs, and fats. But simultaneously, as an endocrine gland, it has specific cell clusters called the islets of Langerhans. These clusters secrete hormones directly into your bloodstream, specifically insulin, which works to lower your blood glucose, and glucagon, which works to raise it. Finally, we arrive at Section 6, Digestive System Disorders, basically looking at the clinical realities of what happens when these intricate systems are disrupted. Remember that protective mucus layer in the stomach we talked about? Well, when that fails, the harsh hydrochloric acid can actually etch right into the tissue, creating painful sores known as peptic ulcers. This is very often caused by an infection from a bacterium called H. pylori, but it can also be triggered by excessive aspirin use or severe stress. We also face threats from the outside, like food poisoning. This occurs when we ingest preformed toxins from bacteria like Staphylococcus found in contaminated food. The chapter also outlines other severe systemic and habit disorders such as dyspepsia, obesity, where BMI exceeds 30, and severe eating disorders with deep psychological roots like anorexia nervosa and bulimia nervosa. So, we have journeyed all the way from how a microscopic, single-celled amoeba eats to the vast, highly regulated, multi-organ factory of human digestion. It's a lot to take in. So, I want to leave you with this final, thought-provoking question. Considering the incredibly intricate chemical and mechanical processes occurring from your mouth all the way down to your intestines, how might your daily food choices be altering the delicate balance of your own holozoic nutrition? Think about that incredible biological machinery operating inside you right now. Thank you so much for joining me in this masterclass explainer. Keep learning, keep questioning, and I will see you all next time.